Hello, everyone. Hope you're all doing well. My name is Allison Zamudio. That's spelled with two L's and last name Audio, but with a Z and an M. As you can see, that's how my name is spelled and today's date. All right, now that introductions are out of the way, let's begin our talk of case one on pyruvate kinase and how its isoenzymes have some correlation to cancer cells. Pyruvate kinase is the final step of glycolysis, which is a process of breaking down glucose to make energy. Specifically, it converts phosphoenolpyruvate, or PEP, into pyruvate so one molecule of ATP can be made, or is made. What is interesting is that pyruvate kinase has different isoenzymes based on the type of tissue it is found in. In the liver and kidneys, it is as type L. In erythrocytes, or red blood cells, it is expressed as type R. It is expressed as type M1 in tissues that need a lot of energy, so the brain and the muscles, and as type M2, which is used in regenerating cell types like embryonic or stem cells. PK is like a traffic light or so under normal conditions, where it handles how fast or slow everything moves to make energy. This leads to how it is regulated through three regulations: through allosterically, through, you could say, covalent regulation, I would say phosphorylation for the most part, and or hormonal factors. The allosteric activator of PK is fructose 1,6-biphosphate, which activates PKM2 by stabilizing its dimer conversion to active tetramers. What this means is that when a cell wants to speed up the process to fill the cell with energy, the activator works as a green light to activate PK and allow the glycolysis process to follow all the way up to its ATP generating step, so the last step of glycolysis. The same could be said when the cell is full of energy, or lots of ATP. It is regulated by several amino acids. Alanine specifically acts as an allosteric inhibitor, or red light, to slow or stop PK activation. Then there is covalent regulation, which is specific to the liver. Sorry. If you don't eat, your blood sugar drops. So glucagon will signal the liver to make more glucose. Human liver PK is regulated through phosphorylation, which occurs on serine 12 on the N terminus to interrupt interaction with the body of a protein, which means that phosphorylation of amino acid residues also have a type of allosteric regulation on the PK on whether it is activated or inhibited, along with other types of kinases. And lastly, there is regulation by hormone factors or transcription factors, if you will. Hormones such as insulin can increase the transcription of hepatic L type PK or be inhibited by glucagon. That in total leads to how it can be thought of as counterintuitive when you go into regards of cancer cells or so. Cancer cells, they can be thought of as like heterotrophs, where they rely on other organisms for their energy, which is why it prefers glycolysis. Yet, it does not actually use PKM1, the isoform that is always demanding energy, but instead it uses PKM2. That is because its less active dimer or enzymatic activity leads to a buildup of glycolytic intermediates that rewire the metabolic pathways and move them toward biosynthesis of cancer cells. So cancer cells, they are not necessarily trying to reproduce, but they're trying to blocks of make building blocks of cancer growth. And the thing is that PKM2 can also move toward the nucleus, so it can promote growth also. That's all I have for case one, and now that was a lot of facts to present. Now I will demonstrate my ability to answer questions presented in a medical situation for case two. Let's say I have a young female patient that has a deficiency of muscle glycogen phosphorylase and has a set of questions for me to answer. Hmm? The first question is, I'm not sure I understand the issue. Can you explain how muscle glycogen is used normally during exercise? That is an excellent first question. You see, when you eat, your muscles store a type of carbohydrate called glycogen, which contains glucose that provides energy to the body. When you begin exercising, an enzyme called muscle glycogen phosphorylase comes and breaks that glycogen stored into glucose 1 phosphate, which is another form of the glucose molecule. That molecule converts into glucose 6 phosphatase, which is an enzyme that goes through glycolysis to make ATP, the energy needed for your body. ATP is energy needed to make your muscles contract, flex, or release. And lactate is also produced in the body. Your deficiency means that your muscles can't break down the glycogen needed to allow your muscles to move the way they normally would and results in muscle weakness. Her second question was, I really like to take long walks. Will I still be able to do this with my friends? Yes. You can still take long walks with your friends since your muscles are not using glycogen for energy, just oxygen. I would advise that these walks should be on level ground and not like climbing up stairs, as that will tire you out more. Her third question, as a result of this deficiency, will I need to get up at night to eat to maintain my blood glucose levels? A midnight snack is not necessary to maintain your blood glucose levels, though I personally enjoy it just for the taste. Your blood glucose levels are dependent on the glucose in your liver, not in your muscles. And finally, do I need to worry about producing excessive lactate during intense anaerobic exercise? Not at all. You would have no or minimal production of lactate. Since your muscles can't break down glycogen, less glucose 6 less glucose 6 phosphate enters glycolysis, so there are low levels of lactate. However, this means that you would experience fatigue easier or muscle cramps when you do intense workouts. I hope this answered all your questions. Do not worry, it may mean having to adjust your exercise routine a little, but there's nothing stopping you from leading a normal life. Have a good day. Okay, I hope that is everything well. I hope you guys are doing great and hope everything's going to go well for this semester. Thank you. Bye.